Hey guys, welcome back. Today we're just going to do the last lecture on active transport. You're really only going to be responsible for the definition of active transport, but I am going to teach you a little couple extra things just because I think they're cool and interesting. And later on in the year, you may see something that we're going to learn in another topic or a video I may show you where I can relate this material that I'm about to teach you back to the actual content we're on at that point. So, active transport, what does it mean when you're active? Probably moving around a bunch, doing all that kind of things. So, active transport is there are other necessary molecules or nutrients that are needed by the cell, but they're already found inside the cell in high concentrations. So you're going to need a little bit of energy to move this molecule into the cell because you kind of have to force it into the cell because you're going against the concentration rule. You're going against the fact that things move high to low. In this instance, for active transport, you're going from low to high. Okay, so it requires energy in the form of ATP. So if you were to think about it, walking in New York City, if you walked against the way people, the vast majority of people are walking on the sidewalk, it's going to be tougher to get between people. You're going to have to force your way through the crowd, and it's going to be a little more difficult. So, types of active transport, you've got pumps. You have exocytosis, which is moving things out. You have endocytosis, which is moving things in. You have pinocytosis, which is moving things in and out through pinching. You have phagocytosis, which is moving things in and out by engulfing. And you have a contractile vacuole, which I'm going to talk about specifically in simple-celled organisms. So endocytosis, endo meaning inside and pino meaning pinch, is the process by which cells surrounds and takes in material from its environment. So if you look at the center image, okay, right here, we're looking right there, okay, you're going to see that these objects are getting, this yellow stuff is the cell membrane, they're getting pinched. You see how there's a nice little bubble forming around the object that we want? Okay, that's going to eventually keep pinching, 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 and you're going to end up with this kind of cell right here inside the cell. I think it's really cool. We're going to cover phagocytosis in another couple slides, so don't worry about that right now. Well, first slide, I should say. But phagocytosis is engulfing. If you guys ever watched Ghostbusters, I think the Marshmallow Man, he could like absorb things or kind of engulf things. Now, this is kind of the idea that like the blob is just going to go over to the object and completely surround it and then ingest it. This actually is done by our white blood cells when we have an invader and our white blood cells attack, or simple-celled organisms like amoebas. Then you have exocytosis. Exo means exiting, moving. So this is the expulsion or exiting or secretion of materials from the cell outward. Okay, so sometimes those things that are moving outwards are hormones, which are good things leaving the cell to send a message somewhere else in the body. And then sometimes they're waste products that you just want to get out of that cell. But the cell is going to bring them in a tiny little bubble like this that we call a vesicle close to the membrane. It's going to attach itself to the membrane and it's going to open and release all the stuff and everything's going to go out all happy and whatever. Okay, but look at the opposite. So I actually put these images next to one another because on the left hand side you have exocytosis, things moving out of the cell, and you have endocytosis, process of taking things inside the cell through pinocytosis or phagocytosis, something. Lastly, we have contractile vacuoles. Now, this is something specific of single-celled organisms, specifically protists that live in fresh water. If you think about it, their environment is very high concentration of water molecules outside of the cell, and inside the cell you have a lower concentration. So water is going to generally want to move into the cell. So in order to prevent it from bursting, you actually have these contractile vacuoles where the vacuole will fill up with water. So if you're looking at this image right here, that little bubble right there, that's a vacuole filled with water. Versus when it gets squeezed, here's the same vacuole, but it's empty. So it's going to squeeze the water back out so it can keep that happy homeostasis water level inside that simple-celled organism. Okay, main function for that is to prevent it from bursting. Hope this lecture helped. If you guys have any questions on the whole entire cell transport topic, please let me know.